And for particle exposures, he did a couple of different scenarios. One, he put particles in the ovalbumin that he was using for sensitization. Another time, he basically just did inhalation exposures. And what he did in doing these studies is this was over a 6-day period. Then he waited for a period of time before he then challenged these mice with an aerosol of ovalbumin to see what kind of allergic response that they would have. This is a classical model for allergic sensitization and challenge. So in doing this type of study, Chris decided that he would look only after the second sensitization or allergen challenge. He would look at cells that he could recover from the lung by bronchoalveolar lavage or valve. He would also look at the blood as a systemic marker, and then he would also look at the lung for changes that might be occurring there. So what I've shown here are just one cell type that he recovered from the lungs by using bronchoalveolar lavage. And what I wanted to show here is that he, in creating different approaches in doing this, is actually looking at, this is the control animals here. This black bar would just be ultrafine particles alone. Here is just the ovalbumin by itself. This blue bar is the ovalbumin with instilled ultrafine particles. This is ovalbumin with inhalation of the particles, and then, sorry. And then this last one, oh, I'm sorry. It's, yes. And then this is basically, the last green bars is really just simply showing instilled and inhaled ultrafine particles with ovalbumin. And he sees a significant effect with this combination of particle exposure during sensitization and with inhalation of the particles. These are just showing eosinophils, which are also known to be an important cell in allergic responses. These they're still all the same controls and exposures here, but the green bar is just simply showing the presence of a trend for elevation in eosinophils. This is not statistically significant, but again, it's associated with that particle exposure that he wouldn't see. He doesn't see any elevation at all if he's just exposing them to particles alone, if the animals are not sensitized. Yes.
So the term that we need, and that is, I think we all understand inhale. What about instill or installation?
The installation, that is actually, when he does that, he's actually putting the particles in by intranasal introduction of the particle with the ovalbumin. So by installation, he's referring to intranasal installation. These mice will, when they're lightly anesthetized, they'll spontaneously sniff anything that basically is presented to them at the nares of their nose.
How is that different from inhalation?
From inhalation?
Just be physically, go ahead.
Yeah. With the installation of the particles, this is where he wants to put the particle associated with the allergen. So he wants a simultaneous exposure that occurs during that time. By inhalation, that is actually done after he does the installation of the ovalbumin. So it's a timing difference, is the critical part of that. In looking at circulating antibodies, because this would be an important part of this, he's looked at ova-specific IgG1. This is just a general antibody, and the bottom line here is that in looking at this, he sees that there's a general trend for increased levels of circulating antibodies, the IgG1 antibody, with the combination of both installation and inhaled ultrafine particles with ovalbumin sensitization. It's not statistically significant, but it's kind of giving us a pattern of what's going on. Looking at total IgE, this is actually more of a marker for the sensitization. I refer you to the last two bars on the right-hand side of this graph, which the red bar is showing the ova with inhaled ultrafine particles, or the green bar, which is the ova with the instilled and inhaled ultrafine particles. And these are both significantly different compared to the control or to just inhaled ultrafine particles. This next bar graph is just for the histology of the lung. One of the important things about sensitization in an asthmatic-like condition is the increase in the amount of mucus that is present within the lungs. And he measured this as mucous substances of epithelials found within epithelial cells of the conducting airway, the proximal airway. And compared to the green bar, which is the combination of instilled and inhaled ultrafine particles, there's a significant difference that this is significantly elevated above any of the other conditions, again suggesting that perhaps sensitization and the inhalation of particles can be very important. And this is simply a light micrograph to show on the left a control lung, on the right side an animal that actually has been exposed to the old albumin in addition to the inhaled ultrafine particles. So one of our aims was to test if inhalation of San Joaquin Valley air, concentrated ambient particles, would differentially increase allergic airway inflammation. So this was a study that was done at the exact same time that Laurel did her study with the progressive 12-day study. But in this instance, Chris was only working with his mice during the last 6 days of that particle exposure that Laurel described. This is just simply showing the station as well as the chambers where the mice are housed. For doing this study, this again was done in which the animals, or the mice, were exposed to concentrated ambient particles on the Shaw Avenue site for 6 hours a day, and on days 2, 4, and 6, they were intranasally had installation of just the ovalbumin, and so particles were only by inhalation. Then there was a period of time of waiting before he actually did 3 OVA challenges that were done separated by 24 hours. And after the end, 24 hours after the 3rd allergen challenge, he then went through the procedure of looking at cells recovered by lavage, looking at blood components, as well as lung histology. And looking at the inflammatory profile, this is actually just showing the total numbers of cells recovered by lavage from animals that filtered air actually are still animals that are sensitized and challenged to the ovalbumin. The yellow to orange bar is actually representing animals that were exposed to the fine and ultrafine particles, or the caps, within the Shaw Avenue site. There's just a trend that is not significantly different. What is interesting is if we look at further and break down the inflammatory cell profile, that there is a significant elevation in neutrophils, in lymphocytes, and total granulocytes for those animals exposed to the concentrated ambient particles in comparison to those animals that were just exposed to the filtered air on the site. Eosinophils shows a trend for elevation with particle exposure, but is not statistically significant. Circulating antibodies, the IgG1, shows a trend for elevation with CAPS exposure compared to the filtered air sensitized animals, but it's not statistically significant. And for total IgE, we saw no differences between particle exposure and those animals that were exposed just to filtered air. In terms of pulmonary cytokine responses, looking at what was happening in the lungs, this again is showing, remember Th2 is your allergic response, Th1 would be more of a pro-inflammatory response. What is interesting here is for those animals, again, blue would be just the filtered air sensitized animals. The yellow middle bar, yellow-orange, is the animals exposed to the concentrated particles that are sensitized, and then the gray bar, the 3rd bar, is just the sham animals. And there is a significant elevation in